Hi class, welcome to lecture number 16. This is the last lecture of the semester. I'm sure you're just as sad about this as I am, but it's been a very challenging semester. It's kind of, I'm happy and I'm sad at the same time to give my last voiceover, and I'm sure you're very happy to hear it. So let's go ahead and dive in and not waste any more time. This lecture focuses on I think it's chapter 20 of the textbook on antimicrobial medications and antibiotics and how they work and aspects to how we use them in care and how they interact with microbes. So the development of antimicrobials and antibiotics are actually it's really interesting because a lot of times it was found fortuitously, but then at the same time there were scientists, usually chemists, that did crazy amounts of experimentation to figure out and basically screen really large quantities of different kinds of chemicals to figure out if they were effective or not. So there was some serendipity, but there was also a lot of really hard work, which is basically the definition of science. There's some really kind of lucky discoveries on the one hand, but then there's also those scientists that grind it out and make those serendipitous discoveries work for the masses. So the first scientist that was really credited with finding an antimicrobial substance is Paul Ehrlich. I want to say that he was I think he was German. He was a scientist, and he found that he noticed that when he would apply stains to cells, that sometimes bacteria took up these stains when mammalian cells or other cells didn't. In fact, there's a bacterial stain that we didn't do in lab. It's called the Ehrlich stain that is named for him. And so he started searching for so of course, when you see basically bacterial cells differentially responding to a treatment that mammalian cells don't, as a scientist or as a physician scientist, you're thinking, okay, maybe this can have some kind of therapeutic effect. Maybe I can find a chemical that has some effect on bacteria that doesn't have that effect on mammalian cells. And so he screened all of these different, I want to say he focused mostly on arsenic derivatives because he knew that arsenic was an effective treatment against bacteria. And he wanted to see if it would be effective against syphilis. And again, syphilis, no one really thinks about syphilis anymore because we basically found the cure, and it's more or less eradicated in many parts of the world. But at the time, roughly 100 years ago, syphilis was a devastating disease, and it was one of the major causes of death. And it had been around for ages, and it was a terrible death. It was really painful, and in the tertiary stages, in the later stages of this disease, you basically lose your mind, and it's a terrible way to die. It's a terrible disease, and in fact, just to kind of underscore how bad it was. What some physicians would do, so the bacteria that causes syphilis is called Treponema pallidum, and it is a bacteria that likes slightly cooler temperatures than our body temperature. So it's not a true mesophile, and this is why it actually. Am I thinking about the right? Yeah. And so one of the treatments for people with syphilis is they would actually give them malaria. Because malaria gives you crazy high fevers and for a very sustained period of time. And so if you can only imagine, in your mind how bad syphilis must be in order to give yourself malaria on purpose to treat syphilis, because there was no cure for it, right? So he found this arsenic derivative, and he named it salvarsan. And I want to say it's some mishmash of like savior and arsenic or something. I forget the two words that he came together, but it's an arsenic derivative that was effective against syphilis, but it was also very, very toxic to people. So that was kind of the first that he was credited with really having the first antimicrobial chemical, and it's called salvarsan. On the other hand, there was another scientist who was looking at different kinds of dyes, and he saw that there was a dye called Prontosil that when injected into, I want to say he injected it into rabbits, only in vitro. So it didn't work. It's interesting. It didn't work in a test tube, but it worked in a mammal. And again, it was either mice or rabbits, I forget. Because what happens is we have an enzyme in our bloodstream that cleaves the prontosil dye into a drug called sulfonamide, and it was really the first. It's a part of the sulfa class of antibiotics, and this is very effective. This sulfonamide is very effective against streptococcal infections, and he. I'm trying to remember the story. There's kind of a cool story. Was it this one? Oh, ouch. Hold on 1 second. I gotta pause. Something's biting me. Okay, I remember what I was going to say. Now, Prontosil, or the derivative of Prontosil, sulfonamide, and Salvarsan are what we're going to call antimicrobials. And this is really important because antimicrobials are chemicals, right? So these are chemicals that are have been purified and have some type of antimicrobial action. Whereas the next kind of major development was actual products from living things. So we're going to call these antibiotics. So difference between antibiotic and an antimicrobial is an antimicrobial is any substance that broadly works against microbes. In order for it to be an antibiotic, it has to be originally made in a living thing. So a perfect example of this is the first antibiotic, which is penicillin. Of course, everyone knows Alexander Fleming was a scientist that originally discovered the antibiotic effects of penicillin because it came from a mold called Penicillium. And this he noticed that when he grew Penicillium on an agar plate, that all of the, I think he was looking at Staph aureus, basically bacteria that were surrounding that Penicillium, that mold, looked to be almost like it was melting. It was really having this very strong and very obvious antimicrobial effect. But he grew very frustrated. He wasn't a chemist. He was a, what was Alexander Fleming? I forget. He's definitely not a chemist though, because he got frustrated. He basically quit because even though he knew this penicillin substance had really strong antibiotic properties, he couldn't purify it in a lab, and so therefore he couldn't use it to any therapeutic value. But at some point later, I think only a couple years later, a couple of chemists were able to purify penicillin and came up with a way to mass produce it. And it really made all the difference in World War II. Not sure if any of you are history buffs or not, but if you look back on the history of major wars, especially in this country anyway, when you look at the Civil War and World War I, more men, typically men, more men died during those wars from infection than they did from the actual wound or injury itself. So infection kills far more soldiers and people in general than the actual injury itself. This all changed with the discovery of penicillin, and that started with World War II. Another antibiotic was streptomycin. So this is the first antibiotic that is isolated from an actual bacteria. Remember that Penicillium is a mold. It's a fungus, right? It's not a bacteria. But streptomycin is isolated from a bacteria called Streptomyces. I think it was Streptomyces careseus or something. Some weird. Hold on, I got an important phone call. Sorry. So Streptomyces is, along with other bacteria that are found in the soil, typically use these kind of antibiotic mediators to destroy the competition. Remember that bacteria live in kind of succession. They need to find a niche for themselves, and part of finding that niche, especially in the soil environment, is to basically outcompete and or kill your competitors. And so it turns out that many of the bacteria that we've identified that actually make antibiotics, we isolate initially from a soil sample. Who knew? Who knew that the savior of literally millions and millions of human lives would come from a handful of dirt? So Streptomyces is the example for bacteria. Penicillium is the example for mold, and these both produce antibiotics. So it's a derivative from a microbe that has antimicrobial effects. Come on, go forward. There we go. Okay. Another major advance in the development of antimicrobials and antibiotics. So I'm going to call them antimicrobials. They're broadly, again, I think the difference between what is considered antimicrobial and antibiotic isn't terribly important. I tend to use the words interchangeably, but I just wanted to clarify that because I know that some physicians and some, this will be a big thing when you take pharmacology, which is a big, difficult class for nursing students to take. Okay, so another big advance in the development of antimicrobials or antibiotics is the ability of chemists to alter the structure of the antibiotics themselves to give rise to new, not only new drugs, but those drugs have new properties now. For example, and so most modern drugs, many of the many drugs that are still in use today are really just penicillin derivatives. A good example of this is penicillin G. If you alter it slightly, you make ampicillin. Right here. And this is this change, again, just a very, very modest change in the molecular structure, took penicillin G, which I think was really only effective against gram-positive bacteria, and then changing its structure into what we now know of as ampicillin, now has a broader effective range. It's not only effective against gram-positives, but it's also effective against a lot of gram-negatives. Broad spectrum. Great. Okay. So this is, I kind of already covered this one. Again, most of the modern antibiotics or the derivatives come from organisms that were living in the soil. And the way that these are produced, believe it or not, if you recall way back when we grew the bacteria in those flasks in lab, it's not that much different than how we did it then. We basically figure out ideal conditions for growth. We grow that and we grow it in a huge vat, industrial kind of scale vat. And then we grow those, if you remember, bacteria have a lag phase and then a log of exponential growth phase, and then they have that stationary phase. And right in here is where we see those secondary metabolites start to happen, because this is where, if you recall, this is where the bacteria start to get crowded and the bacteria start to realize that they need to start outcompeting their neighbors. And this is where they're going to start to create those antibiotics. They're going to, the transcription, right, for these, because these are protein products. So the transcription for these genes are going to get turned on. And now these bacteria are basically turning into little antibiotic factories. And the goal of industrial production of these bacteria is basically to keep bacteria always here, right? Always in this kind of, zone where they're constantly producing these antibiotics. They can then extract the antibiotic from the media using chemical processes, basically chemical distillation process and purification processes, and then they can either alter it in some way or use it as is. So if you ever get a chance to take organic chemistry, you should definitely take it, because a lot of the purification processes that first of all, it's a great class. It's actually the class itself is okay. The lecture is, it's okay. The lab is fantastic. It's probably my favorite lab. Second favorite, next to micro, of course, is my favorite chemistry lab. You do a lot of really fun purification processes based on the chemical structure of different molecules. And so you learn a lot of interesting techniques in the organic chemistry lab that could apply to the purification of antibiotics. Okay, so now let's talk about how features of antibiotic or antimicrobial drugs. So what kind of characteristics do we want these drugs to have? Well, first of all, we need to define a term, and that term is selective toxicity. And the idea here is that we, of course, know of chemicals that are terribly toxic to bacteria. A perfect example is arsenic. Remember, that was the drug that they screened, or the chemical that they screened, Ehrlich and his colleagues screened to try to develop a therapy against syphilis. And we know that arsenic is going to kill bacteria. The problem is, of course, is that arsenic kills us too. It's just toxic. It's not selectively toxic. It's just plain old toxic. So we need this chemical or this enzyme or this protein or this substance, right? This antimicrobial substance to be selectively toxic. In other words, we need it to pose greater harm to let's just say bacteria, to the bacteria than it does to us. And so the toxicity, so in order to describe the toxicity of a certain drug, we call it the therapeutic index, and a derivative of the therapeutic index is the therapeutic window. And I'll talk about what that is in the next slide. So when we talk about the therapeutic index, we want the therapeutic index to be really high or really big. So let's think about two concepts. First we have the amount. Again, let's just say that the amount of drug in the blood, it's typically done in micrograms of drug per kilogram of body weight is usually how most drugs are administered, right? So you know the patient's weight and you calculate the micrograms of drug needed per kilogram of body weight. And usually, and again, for most drugs, there's kind of a standard of care and these are already calculated for you. So we want this number, this value, to be as low as possible, right? We want the smallest possible amount of drug that we can give the patient to be effective. If nothing else, it's cheap, right? It's cheaper. We want to be able to give a very, very small amount so that banning anything else, it's easier to give, right? We don't need to give a huge amount of chemical to a person. And you can imagine that the higher that amount gets, that basically we're going to start to run into is the time at which that drug is toxic. It starts to become toxic to the person. So we want this value here, the amount of drug at which it's effective, to be as low as possible, but we want the amount at which the drug is toxic to the person, to the patient, to be as high as possible. So a perfect example, of course, would be like water, for example. Water, of course, is not toxic at all, but there is a theoretical amount, actually an actual amount of water at which you can drink and it becomes toxic to you, right? There's a condition called water intoxication that can actually lead to organ failure and death. So any chemical taken in some crazy amount would become toxic to a person. And of course, for a lot of these therapeutic antimicrobials, that amount is pretty low. So we basically have two competing values, right? The higher this is and the lower this is, that means that we're going to have a very high therapeutic index, right? So we're going to have a very big value divided by a very small value. That's going to give us a high therapeutic index. Whereas on the other hand, if we have a very high amount, so the opposite of this, so let me just erase some of this chicken scratching. Whenever I write on the screen moves and so it looks terrible. So now let's imagine that the amount at which the drug is effective is very high, while the amount at which the drug is toxic is very low. So now we're going to divide a small number by a bigger number, and that's going to give us a low index. And that's not what we want, right? We want this therapeutic index to be high. This is good, this is bad. And so when we have, and there's plenty of drugs that have a very low therapeutic index, but for various reasons, that it's the only drug that, there's a whole host of reasons why the physician has chosen to use that drug. And that drug would just simply require a much more closer monitoring effort by the care staff that is taking care of that patient. Okay, so let's ignore the graph on the left for now, and let's think about the therapeutic index in another way. So we have two terms that I want to discuss first, ED50 and TD50. So ED50 stands for the effective dose. So let's just look at the top one right now. The ED, the effective dose for this particular medication can be found here. It's the effective dose at which 50% of patients are getting that desired therapeutic effect. On the other hand, we have the TD50, which I believe stands for toxic dose. Toxicity dose or toxic dose. Toxic dose 50, where 50% of the patients have an unwanted side effect. So again, we want that value to be as far apart as possible. So we want a big therapeutic index, or in this case, if we're graphing it, we want a wide therapeutic index. Now, the closer these two things move together, in this case, we can see that this is another antibiotic or another type of medication. We can see here that the effective dose didn't really change that much, as long as the x-axis is the same anyway. But we can see that the toxic dose went way down. In other words, a lower amount of this particular medication causes some type of side effect in half of patients. So now our therapeutic index is very small or very narrow. So an extension of the therapeutic index is called the therapeutic window. And the therapeutic window is based on the therapeutic index, but we also have a function of time included in this as well. So if we look here, either one of these, if we take, this top, this top graph, whoops, why did I do that? Sorry, okay. We take this top graph and turn it on its side. Notice that this is anything below this value is subtherapeutic. In other words, we're not going to see the effect. Anything above this window is toxic, right? So again, if we were to take this, I know this is a bit confusing to explain over a video, but if we were to take this graph here and turn it, rotate it counterclockwise, you can see where the therapeutic index is. See, this is our therapeutic index right now. Because anything above this is toxic, anything below this doesn't work, right? So there's our index. Now let's include a time aspect to this. So if we give the drug to the patient at time 0, we can't have, we of course want the concentration, the concentration of the drug in the blood to be above that therapeutic index, right? Our ED50, and it can't peak above the toxic, above the TD50. And so now the question is, how long does it stay in that therapeutic window? That's the question that we want to ask ourselves. And this has to do with lots of things: patient metabolism, liver function, chemical aspects of the antimicrobial itself. But we basically want that duration of action to be as long as possible within reason, right? We don't want it to last, weeks or months or something. But the longer the period of time that it can stay in so here's our therapeutic index, and the longer it can stay in that therapeutic index is called our therapeutic window. So two kind of very slightly different, your book actually doesn't differentiate between these. I want to say that they call the therapeutic index and the therapeutic window the same thing, but to me they're very different things. Therapeutic index simply has to do with the ED50 compared to the TD50. Whereas the therapeutic window also has to do with the this would be the TD50. Here's the ED50. But now we're adding that element of time where the antimicrobial stays in our blood in that therapeutic window. Okay, other ways that we can categorize antimicrobial drugs, we can do it based on the type of action that it exerts on bacteria. So for example, I think we've talked about these terms. I can't remember if we talked about these or not. But of course I'll reiterate here. Bacteriostatic simply means that they're going to inhibit growth. Inhibit bacterial growth, I should say. So in other words, they greatly retard the ability of bacteria to grow. So they're not going to kill them. They just simply slow down either the rate of growth dramatically, or they stop them from growing. Whereas bactericidal, as of course as that word implies, is directly toxic to cells and directly toxic to bacteria and kills the bacteria. So why would we use one versus the other? Well, bacteriostatic drugs are typically less harmful to the host, but they do rely on host immunity. And typically, bacteriostatic drugs are more than fine. If our immune system gets just a little bit of help in this war, and we already learned how effective our immune system is at kind of battling these bacteria. So if we can just do something to basically hamstring that bacteria, our immune system can do the rest. So we would use bactericidal drugs if that infection is in an area that our immune system can't get to very easily, or if we have a compromised immune system and for whatever reason the host defenses can't control the pathogen. Another way of defining antimicrobial drugs is based on the spectrum of activity. In other words, we can have antimicrobials that are considered narrow spectrum and broad spectrum. So narrow spectrum, as the name implies, works on a very narrow range of organisms. For example, we can have antibiotics that only are effective against gram-positive bacteria or only gram-negative. Some antibiotics are only effective against certain classes of drugs. I am going to go and close my window. I have more noise from my neighbor. I'll be right back. Okay, sorry about that. All right, so narrow versus broad spectrum. So there are some antibiotics or antimicrobials that actually only work against very specific kind of families of bacteria. So we would consider these narrow spectrum. On the other hand, we have broad spectrum antibiotics that, of course, are broadly effective. So why would we use one versus the other? Really, how do I say this? A physician would always prefer to use a narrow spectrum antibiotic because this basically negates the possibility of getting the patient into dysbiosis. Remember that one of the side effects or one of the drawbacks of using antimicrobials or antibiotics in a patient is that you're getting rid of our flora, right? We're getting rid of our microbiome, and that can lead to nosocomial infections like Clostridium difficile, infection of the gut, and so on, which are very deadly. It's a big concern, especially in hospitals right now. So ideally, a physician would always apply a narrow spectrum antibiotic for a particular infection. The problem, of course, is that you have to identify the pathogen. So if you don't know what bacteria is causing the infection, you can't use a narrow spectrum antibiotic against it until you know what it is. So on the other hand, broad spectrum antibiotics are a bit sketch to use because they can disrupt the normal flora. But if you don't know the source of the bacterial infection, you have the best chance of effectively treating that unknown bacteria. This is why microbiology is so important, right? They take a sample of that infection, wherever that infection might be, and even if it's just doing a gram stain, even if it's just kind of shooting that off to the lab, and within an hour, right, if they have a rush order on that diagnosis, they can do a gram stain and give them a conclusive diagnosis of whether or not it is even just gram positive or gram negative, and that might allow the physician to make a better informed choice about which antibiotic they end up using. Now, this is on top of all of the other issues that come along with giving patients drugs in general, right? There are allergic reactions. For example, I'm allergic to sulfa drugs. I can't take any of the sulfa based antibiotics. I'm allergic to them. There's also toxic effects. Typically with antibiotics, it's usually some type of nephrotoxic effect. They can be hard on the kidneys. And then, of course, there's suppression of the normal flora. And then anytime you're using an antibiotic, you run the risk of creating antimicrobial resistance, not only in the patient, but in the population as a whole. Come on, go forward. There we go. Okay. So how do antibiotics actually work? Like, what do they do to the bacteria to hamstring that bacteria or kill that bacteria? So we're going to talk about 4 major mechanisms. We're going to talk about cell wall inhibition, inhibition of protein synthesis, inhibition of nucleic acid synthesis briefly. I also want to point out the antifolates work on some metabolic pathways. And then another one that your book mentions is that there's a class of antibiotics that mess with the cell membrane integrity. I'm not going to give any examples or talk about this one. I just kind of wanted to include it here in the list because your book does talk about it. Okay, so let's talk about cell wall first, cell wall synthesis. So remember that bacteria have this really unique chemical in their cell wall called peptidoglycan. We talked about it in the first week of class. And there are many antimicrobials that selectively interfere with peptidoglycan. These drugs have the big advantage to these drugs is that they have a really high or really big therapeutic index. Why would that be? Anybody? I'm getting silence. I'm just kidding. Okay, so why would we have a high therapeutic index for something that attacks peptidoglycan? Well, remember that the therapeutic index is based on the effective dose versus the toxic dose. Would something that attacks peptidoglycan be toxic to humans? No, not at all. We could care less about the toxicity of peptidoglycan targeting drugs because we don't have peptidoglycan. So the big advantage to this class of drugs is that we can take them without having any type of side effects or cross reactivity with our own cells. So there are 3 different kind of major classes or major groupings that I want to point out. There's the beta-lactam drugs. Penicillin is the perfect example of a beta-lactam type of drugs. Another type of drug is called vancomycin, and one that you've probably heard about is bacitracin, which is a topical ointment. So let's talk about these lactam drugs first. So the beta-lactam drugs, such as penicillin, prevent I think there might be a better picture on this one. So they basically interfere with the formation of these peptide chains. If you recall, the NAG and NAM repeats that make up the peptidoglycan cell wall are held together by these very short kind of peptide bridges. And the beta-lactam drugs get in there and they interfere with those peptide bridges. And so they prevent those bridges from forming. So it can't dissolve directly an already intact cell wall, but it can prevent it from growing. So penicillin and beta-lactam drugs in general are really good against actively growing bacterial populations. So the beta-lactam drugs have a different varying, depending on the drug that we're talking about. Some of them are more effective against gram-positive, others gram-negative. The big problem with beta-lactam class drugs is that they have kind of an Achilles heel, and that is beta-lactamase. So this is a major mechanism of antibiotic resistance. So if we look at what the major penicillin and hold on. Let me plug in my computer, hold on. Okay, sorry. Okay, so 2 major drug in this class are penicillin and cephalosporin. And all of these beta-lactam drugs contain what's called a lactam ring, and you can see it here as part of its molecular structure. Beta-lactamase is an enzyme that some bacteria can make that cleaves this ring. It basically breaks it open, and this doesn't work. So it takes an active molecule that has that lactam ring and turns it into an inactive molecule. As you can imagine, this is an extremely valuable gene for bacteria to have, and they trade it like crazy. Remember that horizontal gene transfer that bacteria can do? Well, this is like a $100 bill, right? This is even better than $100. It's $1 million, because if bacteria have this beta-lactamase gene, they are now resistant to these beta-lactam class of drugs. Okay, so what do you do? What do you do if you have something that is resistant to the beta lactams? Well, you give them vancomycin. Vancomycin also inhibits cell wall synthesis. It does not, it's not effective against gram-negative bacteria because, and generally speaking, I should say also, gram-negative bacteria are much more resistant to, well, cell wall inhibitor class of antibiotics, but antibiotics in general, because they have that outer membrane that protects the bacteria from so many of these antibiotics. And we'll talk about that. I think it's the last slide in the lecture too. But vancomycin, it's really only good against gram-positives. It inhibits the glycan chain formation. It basically, oh my goodness, what does it do? I want to say that oh, I think I have the picture. Why am I trying to remember here? Vancomycin. Yeah, it binds to the NAM molecule. And then it basically doesn't allow the side chain of NAM to link up to the NAG subunit. So again, not terribly important, the molecular mechanism, but understand that it's distinct from those lactam drugs. Let's see. What else do I want to say about? Oh, so one of the issues with vancomycin, and again, just drugs in general, but this is one that's particular to vancomycin, is that you can't give it you can't give it orally. You can't. It doesn't. Basically, our digestive system can't take it up, and it doesn't make it into the bloodstream. So you have to give it via IV. And unfortunately, there is a lot of vancomycin resistant gram-positives that are starting to pop up as well, because the NAM basically gets slightly altered over time, and it becomes resistant to binding to vancomycin. Our last example of a cell wall kind of targeted antibiotic is bacitracin. You probably know it as Neosporin. The generic, of course, is called bacitracin. Again, it's toxic if taken orally, so you really should only take it topically. There's no bacitracin oral type of drug, but it's kind of cool how it works. It basically prevents, so again, we didn't get into the metabolism of how these NAG and NAM, how should I say, the precursors to build the cell wall, how the bacteria makes them. And it turns out that they import some of the precursors into the cell, and bacitracin prevents, I want to say it's NAM, yeah, right here. So the cell has to import this precursor into, so this would be a gram, actually we don't know if this is gram positive or gram negative. So it has to cross that cell membrane, and it prevents that that NAM subunit from getting across the cell membrane so that it can become part of the cell wall. So basically destroys the integrity of the cell wall for these bacteria. All right. Another major target for antibiotics is protein synthesis. Remember that our ribosomes, eukaryotic ribosomes, are slightly different than prokaryotic ribosomes. Eukaryotic ribosomes, when they come together, we have an 80S ribosome that is structurally distinct than the 70S ribosome that bacteria have. So because this is a different structure, again, that toxicity is going to be low. We can give these antibiotics to a person. It won't affect the person's ribosomes, but it will affect the bacteria's ribosomes. Lots and lots of types of drugs in this class. They all have slightly different mechanisms of action, and I'll talk about a few of them here in the next few slides. So let's talk about the aminoglycosides first. They work by binding to the 30S subunit, and this blocks initiation. So basically the mRNA can't, I want to say it can't get in, or it stops as soon as it gets in. And so as soon as this happens, so so here's the 30S ribosome. And when the 50S comes, and we remember we need to initiate the translation, here's our messenger RNA transcript, and it can't get through. And so the protein is the code is misread, and the protein comes out either incorrect or misshapen in some way. Aminoglycosides are good antibiotics to give alongside So a more common practice in a hospital setting is to not give just one antibiotic to a patient, but to give two. And the idea here is that this is basically going to negate the possibility of creating antibiotic resistance within that person. So if you think about the ability, a random mutation or a random chance occurrence of having a gene that can give you resistance to the beta-lactam drugs, for example, the chances of them also having a mutation or a gene that gives them resistance to aminoglycosides is nearly impossible. So it's cutting down on the possibility of having resistance, but of course you can imagine that the toxicity and all of those other kind of considerations with toxicity are going to go up. All right, the tetracycline, and most everyone's heard of tetracycline. It's one of the most commonly prescribed antibiotics. This class of drugs binds to the 30S subunit as well, but it blocks the ability of, it basically doesn't allow the transfer RNA to attach. So even though it's attaching, even though the tetracycline, the chemical itself, the molecule attaches to the 30S, it doesn't allow the tRNA that comes in the 50S, it doesn't allow it to come into the, oh goodness, I think it's the E site. No, A site. It doesn't, remember there was the A and the P and the E site and in the ribosomes that allow the transfer RNAs to come in, and it doesn't allow that to happen. And this is going to prevent elongation of the polypeptide. The problem with tetracycline is that it is only effective against certain gram positives and gram negatives. So it's a narrow spectrum antibiotic. The last ones are the macrolides. These bind to the 50S. So in contrast to the in contrast to the aminoglycosides and the tetracyclines, these bind to the 50S subunit, and they prevent elongation of the polypeptide as well. They are, I don't know if I consider these narrow spectrum or broad spectrum. They're somewhere in the middle. Again, they're broadly effective against gram positive. This is true in general of antibiotics, and is a commonly prescribed drug if the patient is allergic to penicillin. You've probably heard of erythromycin. It's probably the most commonly prescribed class of macrolides. All right, this is our third. So we've talked about cell wall synthesis inhibition or cell wall degradation. We've talked about protein synthesis inhibition. And then our third mechanism of action for antibiotics is DNA replication. So the one that I wanted to point out were the fluoroquinolones. These basically inhibit the activity of a protein involved in DNA replication called gyrase. And gyrase is where? Right here. It's this donut-shaped enzyme. And it basically unwinds the DNA. Remember that DNA is a double helix. In order for it to become single-stranded, we have to basically unscrew that twist in the DNA. And fluoroquinolones negate the ability of DNA gyrase to do that. Advantage is that it's broad spectrum. You've probably heard of ciprofloxacin. It's a very commonly prescribed class of fluoroquinolone. The problem is that the gyrase the bacteria becomes resistant to these quite easily, because what happens is small mutations accumulate in the gene that codes for gyrase, and very small changes in the protein sequence of gyrase makes it alters the gyrase enough that the antibiotic doesn't work anymore. Okay, our last mechanism of action is, I think this one's kind of cool to me. I don't know. I think it's interesting to see. Oh, God, I got another phone call. Hold on. Sorry, let me pause. Okay. All right, so let's describe our last mechanism of action, and that is a class of drugs that are folate inhibitors. So in order to understand why this drug is important, we have to understand what folate is, or folic acid. So folic acid is a important precursor to that's involved in the synthesis of nucleic acids. If you look here, thymine, guanine, adenine. These are nucleotides. Remember that it's the T and G and A of the nucleotides. And in order to synthesize these nucleotides, we need folic acid. And PABA is an important vitamin that is involved as a cofactor, coenzyme in this metabolic pathway. Now, humans can't, we don't have this metabolic pathway. We can't make folic acid, and therefore we have to get our folic acid from our diet. So we really don't care if we get folic acid inhibitors or not. However, bacteria make them themselves. And so there are one class of drugs is called the sulfa-based drugs, the sulfonamides, and then there's another drug called trimethoprim that block different enzymes that are important in this metabolic pathway. And so the sulfa drugs do this by mimicking basically, so if you look, here is the PABA structure of PABA, and then sulfanilamide, which is a sulfa class drug, it looks very close. All right, the only difference is in the side chain, the chemical structure of the side chain, and so it looks enough like it to inhibit this first step of the metabolic pathway. So again, low toxicity to people because we don't care about this metabolic pathway because we don't have it anyway. But this is really, basically this prevents the ability of a bacteria to make nucleotides. And if it can't make nucleotides, it basically can't get out of, it can't create new DNA to replicate. So how do we determine, I've been talking about broad spectrum, narrow spectrum, this antibiotic is effective against gram-positives, but not gram-negatives, and so on. How do we know this, right? So in the past, what physicians had done is they would just try different drugs until they got a favorable response. And of course, that's not a very good approach. So a better approach is to determine the susceptibility first and then prescribe a drug that acts against that particular organism. This is why it's always better to use, ideally use narrow spectrum drugs because it's effective against that particular bacteria. It doesn't affect other organisms. So one method of determining susceptibility is through determining what's called the MIC. The MIC is the minimum inhibitory concentration. So if we look here, what we can do is we have three different organisms, organism A, organism B, and C. And we have decreasing concentration of some drug, does it even say? I don't think it, yeah, some antimicrobial drug X. So we can see that at some concentration, the bacteria is going to drop below the minimum inhibitory concentration. So at a concentration of 0.12 micrograms per milliliter, this particular drug X is effective against organism A. Once we have that concentration to 0.06, it's no longer effective. Same approach for organism B and C. We can see that the MIC for B is 1 microgram per milliliter, and for C, it's quite high at 16 micrograms per milliliter. So this drug, this particular antimicrobial drug X, is most effective against A, B, or C? A, because remember, we're looking for the lowest amount of drug that has a therapeutic effect. And in this case, we're looking for, do we know if this is bactericidal or bacteriostatic? We have no idea, right? All we know is that the bacteria aren't growing. So we don't know if it's directly killing the bacteria or preventing them from growing. Another approach to determining susceptibility is the Kirby-Bauer disc diffusion method. So you basically take an agar plate of Mueller-Hinton agar. It's a special type of agar that is poured in a particular way, and you place on the surface. So first you streak the plate with the bacteria that you're looking to determine susceptibility, and then you put different little paper discs on the surface of the agar, and the paper discs are infused with different kinds of antibiotics. And so the zone of clearing, you can see that the bacteria is inhibited from growing around, right? So here we can see a zone of clearance where the bacteria can't grow close to that paper disc because that's where the antibiotic is. So you can see that these two drugs are not effective against whatever this particular bacteria is. I don't want to spend too much time on this because this is actually what our lab is on. I'm going to talk a little bit more about it in lab. So I'm going to kind of skip through this slide and give you that information in the lab. Okay, so how can bacteria be resistant to antimicrobials? Well, there's different mechanisms of resistance. So Remember, so when we talk about resistance, let's imagine that we have a pool of bacteria. We're going to treat it with some type of antimicrobial. It's going to kill all of the cells that are sensitive. But remember that all we need is that one cell, for whatever reason, is resistant. And in 20 minutes, that one cell becomes 2. And then in 40 minutes, it becomes 4, and so on. So all we need is for that one bacteria to survive that application of antimicrobial application, and we now have resistance. So what are some mechanisms of resistance? Well, we've already talked about a couple of them. First is enzymes that can directly inactivate the drug. So we talked about the beta-lactam rings and the beta-lactam class drugs. And if a bacteria contains beta-lactamase, it can cleave that lactam ring of the penicillin class of antibiotics. Another common issue I mentioned, it was very common with the topoisomerase inhibitors. They can, and there's some of the, many of the antibiotics that target the ribosomes are also susceptible to this, but basically the target molecule, the thing that they bind to, changes in some small way such that that molecule can no longer bind to it. So very, very small changes in the 30S ribosome or the 50S ribosome can now confer resistance to for that bacteria in response to that particular antibiotic. So this is this is very similar to a figure in your book. Let me find out what figure it is. Figure 20.14, very similar to that figure in your book, and it talks about the different mechanisms of resistance. Remember, in a non-resistant cell, in other words, a sensitive cell, the drug gets into the cell, it binds to the target, and usually inhibits that target in some way. In a resistant cell, there's basically 4 different possibilities that can happen. So we talked about one. Generally speaking, gram negatives are really good at this. Basically what happens, actually not only this one, but also this one. So gram negatives, in fact, I would say gram negatives are better at this one. So essentially what happens here is the drug can't get past it. Basically there's no uptake. The pores that are the gates, remember there's special holes in the cell membrane and the cell wall of bacteria that only allow certain chemicals to get in. So it's possible that that particular bacteria just doesn't allow it to get into the cell. Another possibility, and a more common possibility, especially in gram negatives, is that the drug can get in, but then the cell kicks it right back out again. Again, this is a classic. This is basically why gram negatives are so tricky to treat, because the drug gets in, but the bacteria just kicks it right back out again. So we already discussed how small alterations in the target molecule can lead to non-binding. So it's possible that the drug can get into the bacteria, but again, the target, like topoisomerase, the 30S ribosome, the 50S ribosome, they change their structure slightly so that the drug can no longer bind. And then finally, we talked about some drug inactivating enzymes like beta-lactamase, that can basically modify the drug itself and thus inactivate it. So all of these can be broadly categorized into 2 different mechanisms of acquisition itself. So it could be due to spontaneous mutation. So in the case of changing the topoisomerase or the 30S ribosome, very small mutations that build up over time could lead to alteration of that target molecule. And this is why it's important to take the recommended dose and take it for the full cycle, because you don't want to give the bacteria a chance to kind of keep replicating in the presence of that bacteria, in the presence of that antibiotic, because the more replication cycles you have, the more mutations you're going to accumulate and the more likely you're going to get resistance. So this is vertical, right? So remember that mutations that are passed from basically parent to offspring are called vertical mutations, or vertical, I don't like the word evolution, but I guess it is genetic changes, right? So it's a vertical gene transfer, or we can get new genes. So horizontal transfer through either a plasmid, through transformation, conjugation, and so on. So I just wanted to quick give a couple of mentions. There's actually a nice table in your book that talks about mechanisms of or not mechanisms, but different concerns that are kind of present in the medical community about resistance to antimicrobials. It's table 20.2. A couple that I just want to point out are the enterococci. The issue with enterococci is that many of them are becoming resistant to vancomycin. Vancomycin is considered an antibiotic of last resort. It's really what you give a patient when there's not too many other options. And with these enterococci that are becoming resistant to vancomycin, it's a big concern because they are becoming resistant. Another one that I meant to put on here and I forgot is, believe it or not, is Neisseria gonorrhea. There are many strains popping up in different, fortunately not too many in the United States, but around the world there are lots of strains of Neisseria that are popping up that are very strongly antibiotic resistant and can be deadly if left untreated, and there's not a lot of treatment options for those patients. Of course, we've all heard of Staph aureus, antibiotic resistant strains of Staph aureus, the MRSA and the VRSA. This is a very common, Staph aureus is one of the more common causes of nosocomial infection through bed sores and disruption of our physical barriers. For a long time, there was an antibiotic called methicillin that was really good at treating Staph aureus infections. However, a lot of Staph aureus species are now resistant to methicillin, and that's what MRSA is called, right? Methicillin resistant Staph aureus. So we've been using vancomycin, and now there are some vancomycin resistant strains of Staph aureus or vancomycin intermediate strains of Staph aureus. And then I wanted to just really point, I think this is the last slide. This is the last slide. And I wanted to point out that an unintended, or I shouldn't say an unintended, something that is of importance here in San Diego, because we have a large population of homeless people that are intravenous drug users and drug abusers. Also with the increased use of not drug abusers, but people with type 2 diabetes, basically people that are giving themselves lots of finger pricks or injections, on a daily basis. A rising threat that is starting to pop up, that there's actually some information on the CDC's website. I can maybe link to it in the lecture page, is endocarditis. And so one of the major causes of death for people that are intravenous drug users and drug abusers, I should say, is a disease called endocarditis, and it's basically a bacterial infection of the sac that surrounds your heart. And this is because we basically are taking bacteria from a infected needle or a dirty needle, and you're bypassing your physical barrier. Remember that a lot of these bacteria can't make it past our physical barrier. Our skin is the best protection that we have against these bacteria, and you're introducing it directly into your bloodstream. And these patients die from bacterial infections, usually of the heart. So a kind of a little known side effect of drug abuse in this country. And again, not only in drug use, but there are more and more therapeutics and medications that people with chronic issues are having, where they have to give themselves some type of injection on a daily basis. So of course they're not abusing drugs, but nevertheless they're still having to inject themselves with something. And what happens here, and I don't know, I'm sure that many of you have experiences with this with your parents, but my mom she's a type 2 diabetic, and she has to give herself these injections. And basically what happens is, over time you stop respecting the fact that this is dangerous, right? Because you give yourself injections and finger pricks every day, sometimes multiple times a day. You start to not, disinfect the area that you're injecting, and you stop caring, you're not scared anymore, basically, of getting an infection. And so stretch that out over weeks, months, and years, and at some point you could get a very, very dangerous infection. All right, that does it for this lecture. This is a long one. I should have cut it into two, but nevertheless, that's it.